Hey everyone, I'm Brook Pyhtila, Director of Content at Addgene, and I'm here to talk about our exciting platform, the AAV Data Hub, a user-friendly place for sharing experimental results with the scientific community. In the almost 20 years since we launched, Addgene has expanded to become much more than just a plasmid repository. As a nonprofit, our mission is to accelerate research and discovery by improving access to useful materials. So our collections grew to include a whole host of plasmid-derived materials, such as pooled libraries, recombinant antibodies, and viral preps. As part of our mission to share information about those materials, we built the AAV Data Hub. Currently, this platform is designed specifically for reports describing experiments using AAV preps distributed through Addgene. We're limiting this to Addgene materials so we can ensure all preps have gone through the same quality control measures, keeping batch-to-batch variations to a minimum and information accurate. Our expert scientific support team can tell you that many scientists have questions about working with AAVs, such as, what serotype will work best for a given neuron or cell type? Why am I not getting expression after injecting my AAV? Why is there expression of my Cre-dependent AAV in non-Cre expressing cells? The Data Hub allows you to find answers to these questions and more, granting access to detailed reports describing AAV experiments and helping you decide which AAV to select or what conditions to use in your experiments. These reports include details that may not be published in other types of publications, either because this level of detail is not included or the expression pattern or results might not have worked as expected. To use the Data Hub, you can filter by serotype, promoter, species, or application. You can also search by the name of the cargo, brain region, or author. Then once you narrow it down, you can click on View Details to see the full report. We recently improved our database and the submission process to make it more robust, flexible, and user-friendly, thanks to support from the Chan Zuckerberg Initiative. We know there's a treasure trove of valuable information sitting in notebooks in labs across the world, inaccessible to scientists looking for answers. That's one of the main problems we want to solve with the Data Hub, but for that, we need your help. So, I'll let Jason Nasse, a senior scientist at Addgene and resident Data Hub expert, tell you how to easily submit your report so you can get that information out and do your part to help the scientific community.
Thanks, Brooke. And thank you for your interest in contributing to the Data Hub. We know just how valuable your time is, so our team at Addgene has worked hard to ensure submitting data is as easy as possible, while still collecting the necessary details for comparing AAV experiments. Your report will include details about the virus and injections, images, and a description of the results, whether positive or negative. One thing to keep in mind is you can save the progress of your report at any time, so there's no need to go through the entire process in one sitting. To get started, navigate to datahub.addgene.org and click the orange Contribute button at the top of the page. If you haven't logged in yet, you'll want to do that now. You can log in using an Addgene account or an existing Google account. Next, pick your report type. As Brooke mentioned, we're currently only accepting AAV reports, but we'll be opening this up to antibodies in the near future. Now follow the prompts to answer questions and create your report. In the first section, you'll provide contributor information such as author names and funding sources, as well as the title and abstract of your report. The abstract should describe your experimental conditions and the general outcome, and can be as short as a few sentences in length. Next, you'll provide virus and injection details. You can see here that these are short answer responses, and we have helpful text to guide you through the expectations. If you're not sure what the actual titer of your virus lot was, just email us at datahub@addgene.org, and we'll look it up for you. For the injection site, we're using standard nomenclature from the Neuronames Ontology website. This ensures consistency in identifying the brain region you injected so that the information can be accurately compared across different reports. We recommend that you also include the stereotaxic coordinates in the additional details box, along with any other details not captured in the previous fields. The next section covers information about your experimental model and how the data was collected. Information about retrograde transduction and functional testing are especially useful to other scientists and would be great to include here if you have it. Now it's time to upload your images. We ask for PNG files with a minimum resolution of 300 dpi. We suggest submitting both a low magnification view of the entire brain region injected, as well as a high magnification view for cellular specificity. Importantly, please add a caption that accurately and specifically describes what is being shown in the image. If you're not sure how to word this, check out our How to Contribute page for more guidance on submitting images with the most impact. Remember, you can save your report at any point along the way to collect more information and preview it before submitting. Once you're ready, you can agree to the terms and submit your report. Reports will be made accessible to other researchers under the Creative Commons Attribution 4.0 International Public License. Now that your report is submitted, Addgene will review the report and work with you to make any adjustments. Once the report is reviewed and approved, it is published and assigned a citable DOI. If you're curious what a finished report looks like, here's an example of one submitted by Dr. Eugene Dimitrov. You can see it's a bit like a mini paper, starting with title, abstract, authors, and funders, followed by some amazing images, and then details about the virus, injection, and subject. Finally, a brief but helpful summary explaining the results of the experiment.
And that's it. Remember, the Data Hub is a community resource and can only succeed if you contribute. Your data has great value to others, and sharing data speeds science.
Contribute now at datahub.addgene.org. And as always, we're here to help. If you have any questions, just email us at datahub@addgene.org.